name is Chelo, and I'd like to tell about the work that we've been doing on how bacterial adaptation can change C. elegans performance and the ability to adapt to new environments. So,
let's go full screen.
There you go. Okay. So we all know that bacteria can have strong effects on hosts, right? So that's one of the reasons that we are very interested in studying them. So some of these effects can be quite deleterious, leading to health issues, eventually to death. And in other cases, it can actually affect development of organisms, and that can have strong consequences throughout their lives. On the other hand, it's obvious that we also know that bacteria sometimes can be quite beneficial. And we know about this, for instance, from the digestion in ruminants, but also they help predation in squids. And for the past few years, there's increasing evidence that bacteria can have strong consequences for human health. Now, in C. elegans, so C. elegans has been used widely as a study system to study the effects of bacteria on hosts. So not only to see how these interactions can evolve, but also to go into the mechanistic and to molecular details of how these interactions can come to be. So once again, some of these effects can be quite deleterious, and they lead to increased mortality of C. elegans, decreased longevity. Sometimes can even go to have problems of degenerative diseases. On the other hand, it's possible that some of these effects can also be beneficial, such that as when we see that on the other hand we have increased longevity, or in some cases you can have protection against infection. For the time being, I will leave fast development from this dichotomous classification, whether it can be good or bad is something that remains to be seen. Now, one of the questions that still it's not clear at which extent this wide range of effects that we see is something that comes about of the particular strain or species that we're looking at, with its unique history of either being a pathogen or mutualistic, or it's something that is much more flexible that depends on the context or the environment, or any particular mutation that that particular bug might have got into. So one example of these was this case where Douglas Browning and collaborators, they used this bacteria that probably all of you that did any cloning in the lab already used. This is a K12 E. coli, MG1655. And what they did is that restored this locus that is otherwise not active. By doing so, now this bacteria produces in this outer layer the O antigen, which is this lipopolysaccharide molecule that is well known and is being known to be a virulence gene in many other cases. So what they see is just by doing so, maintaining the same genetic background, now this bacteria turns quite virulent for C. elegans. And what they see is that it decreased survival by 50%. So this is the survival curve they observed. Over here, what we have is the survival of C. elegans when they feed on E. coli OP50, which is the standard or normal food source for C. elegans. This is what we see, the survival curve of the MG1655. And what we have over here is of this construct that they put the O antigen in the surface, where the survival is much reduced to the extent it's quite similar to one known pathogen of C. elegans, Pseudomonas aeruginosa. So in our lab, we picked up this strain and actually went further on to try to characterize a little bit any other fitness effects. So what we could see by measuring fertility a long time throughout the individual's lifespan, what we see is that, so what we have in here in black is that the fertility of the MG1655, and when you compare it with the white ones, actually you see a reduced fertility, but not only that, you also see a delay in the reproductive time peak, which in this case is about 50%. Naturally, when you also measure the extent to which the first eggs start to be laid on the plates, you also see a delay in those timings and a 65% increase. So we have the same overall genetic background. Now we have the antigen being expressed on the surface, and you do see these remarkable changes. Now, to which extent is this something that we could have easily in bacteria adapting in nature? So could have some kind of ecological or evolutionary relevance. So to test for that, actually we went to another system. So what we decided to do is to use this benign E. coli OP50, once again, regular food source for C. elegans, and adapt it to normal growth conditions of C. elegans in the lab, being 20 degrees of temperature, the nematode growth medium, constant humidity, and we did this in replicate fashion. And we not only use the most standard environment, this nematode growth medium, but also use a new environment which we know is stressful for C. elegans because we have an increase in salt concentration. So we have 20 replicate populations in low salt environments and 20 replicate populations in high salt environment. And we characterize this phenotypically and genetically. But for the purpose of this talk, what I would just like to show you is that basically it's the thickness that we obtain from competitions against ancestrals, because these bacteria carry fluorescent markers and you can easily compete and get frequency change. So what we show over here is their fitness in the low salt environment and plotted against the fitness in the high salt environment. In blue are the populations that adapted to high salt, and in orange the populations that adapted to low salt. So what can we get out of this? So basically they all adapted, values are positive. There tends to be more adaptation in the same environment, in the environment that they were adapting to, meaning there's some kind of specificity, or just to say that salt actually matters over here. Might have some more adaptation in high salt adapted populations when you compare it with the low salt adapted populations. And in the first case, we also have some evidences of possible trade-offs because populations increased their fitness in high salt, but at the cost of a decrease in fitness in low salt. Now, from this, actually, we selected the bacteria that had the higher fitness in their respective environment. And we use this as food source for C. elegans to try to see what would happen to its performance in terms of growth rate, and whether or not that would give us some hints that it would be interesting to use those as food source for a new round of evolution now in C. elegans. So what we have over here, this is growth rate on C. elegans, four different genotypes, measured in low salt and in high salt, and beware of the log scale over here. Okay? So while they are feeding on ancestral E. coli, what we can see is that salt is quite ulterior for C. elegans. They grow much less. And what's interesting to see is that each genotype actually behaves a little bit different. So this interaction is significant, and you have some genotypes that are more affected than other genotypes. For instance, in this case, the CB486756 is actually the one genotype that is less affected by salt. One more interesting point for this talk is that when we change the bacteria that they are feeding on, in this case, you'll be feeding on the bacteria that was adapted to low salt, now suddenly salt has a more tremendous effect, so they grow much less. The rankings, relative rankings of these species, of these genotypes is not much affected, but that could be if we feed them with the bacteria adapted to high salt. So in this case, all of a sudden, N2 strain starts to be the strain that is less affected by salt or low salt overall. Now, this is also good for us because it tells us that there's a genotype of the worm, by genotype of the bacteria, by environment interaction, which tells us that if we had adaptation of C. elegans in these two different environments, depending on the bacteria they would be feeding on, then adaptation could possibly lead to different results. So that's where we went next. We decided to do this experimental evolution on the C. elegans population. So this is a genetically diverse C. elegans. So this is not regular males hermaphrodites, but a male-female dioecious populations, because we wanted to ensure that we have the highest possible recombination and segregation of genetic variability. And an important point over here is that we impose discrete generations at a given time point. So once development ends, we basically kill all the adults and get the embryos and let them constitute the following generations. So this was done for 40 generations, 4 replicates for each of these conditions, and the census sizes is at a carrying capacity at the top level of 10,000 individuals, but it can become a little bit lower, as we will see next. So this is just a reminder to say that we have plenty of bacteria in all those plates. So we tested this by changing amounts of bacteria, and we do not see any differences in terms of growth rate on C. elegans. Now, it is also important to say that bacteria is given de novo, so this is not a coevolution experiment. If you're used to coevolution experiments or coevolution setups, basically this is one of the controls that people use to do. So what I'm plotting here is the growth rates of bacteria of C. elegans while they were adapting to high salt, just for the first 25 populations. What is immediately clear when we measure growth rates of C. elegans feeding on the ancestral in gray, or in the bacteria adapted to high salt, well, in all cases they decrease initially, and they increase the growth rates and start to stabilize, possibly adapting. And interestingly, what happens when they are feeding on bacteria adapted to low salt, and let me make it more clear, is they basically all die. So usually we started with 4 replicates. We replicated these 3 more times and ended up with having 12 populations out of 12 getting extinct in the first 3 generations. Now, something is obviously happening over here, and we're trying to figure out, it's not something which is just because of the bacteria, because if we get the same growth rates, but now for the C. elegans populations adapting to low salt, we do not see any difference in growth rates depending on bacteria. Now, actually, we should not be surprised because we know a little bit about the system. We had seen before that in high salt, C. elegans development is delayed by itself. So this was done with hermaphrodites fertility, and we see that at 72 hours after being hatched as first larva stage, the populations growing in high salt, they are not able to produce as many offspring as the populations growing in low salt, but they are able to catch up later on. And we had other evidence showing that actually there's this developmental delay. Now here I'm plotting the developmental time, and the proxy for this is the time that we start to see eggs appearing on plates for 2 genotypes, N2 and CV4A55, in the ancestral bacteria and the bacteria adapted to low salt. So once again, we see that there's this increase. So what is the model that we have so far to explain our results? Well, basically, this is a distribution of fitness. Affects a long time. This could be fertility, for instance, as I showed you earlier on. And there's a specific time point that we collect individuals to constitute next generations. And this time point does not change for all of our treatments. When you have populations in high salt, what happens is that you have a delay in development, so the distribution is shifted to the right, but most often in the ancestral and the bacteria adapted to high salt, they're still able to maintain the population sizes. Now, when you're feeding on the bacteria adapted to low salt, there's a further delay on their development. And now all of a sudden, these populations are not able to maintain growth rates above 1, so in the end, they become extinct. So I would just end up saying that we have to make conclusions of this work in our system, that bacterial adaptation can delay development, and in the end, in these cases, can lead to population extinctions under stressful conditions. Obviously, what I would like to leave you with is this question that when you go into nature and you have environmental change, to which extent part of the problems is the fact that the new environment has new abiotic conditions, or the fact that populations are adapting, and this itself can be quite stressful for other species. So just a thank you note for people that did most of this work. Ana Paula and Josiane, postdocs in the lab, did most of this. Also Ana Laranjeira, David, research technician, also important contribution of 2 undergrads and different sources of funding. And obviously for you for listening. Thank you.